One of the last chapters that we'll be covering in microbiology is chapter 25 on applied microbiology and food and water safety. And this is a very important chapter. It really encompasses what local public health departments do, especially regarding exposures in food and water, and making sure that they are safe for consumption, and also tracking down infectious diseases that are associated with food and water, which are usually diseases of the gastrointestinal system, which I will be covering later on this semester. So anyhow, this is the second video lecture for Tuesday, the 31st of March, and I will cover this chapter in its entirety in this video lecture. So our objectives are to discuss microorganisms in water and wastewater treatment. Wastewater treatment is very important. It's a part of civics, and every municipality has some type of wastewater treatment. If you have a sewage system, then that sewage system is ending in a wastewater treatment facility. And so we're going to discuss the basics of that. We'll also talk about microorganisms that are used in food production, as well as microorganisms that introduce spoilage. And we'll talk about microbes that make other products. And just jumping right in, let's talk about wastewater treatment. And if you live out in the country, then you may get your water directly from a well. And well water is generally pretty darn pure. So it's been purified already through flowing through sediments over long periods of time. And that sediment has a lot of carbon dioxide in it, which acts as a cleanser. It may have oxygen in it to oxidize any microorganisms. But it's always a good idea to check your wells and make sure that there aren't any organisms like Giardia or Cryptosporidia that cause lower GI disease. And so you can do that. You can have the public health department come out on a yearly basis and check your well water and make sure that you don't have pathogens coming up from your well. Okay. Now, in terms of wastewater treatment itself, first you take the water and you put it in a large reservoir. This is just a large area that is used for sedimentation, and you get rid of the solids, and you also add copper sulfate at small quantities to prevent algae and cyanobacteria growth. If you get algae and cyanobacteria, then you would get eutrophication, and that would lead to anaerobic conditions, which may not be the best conditions for wastewater treatment. Okay. You then go to holding ponds where there's further settling of sludge out of the wastewater, aeration to promote aerobic activity, and then filtration to get rid of solids. So when the water is released back into the environment, and then once it's released, then it won't have large particles in it. And then finally, you may include chemical disinfection. Chlorine gas at about 1 or 2 parts per million is injected directly into the water. Okay, so if you have untreated water, first you have a catch basin, okay, and there will be some sedimentation there. Then it will go into a pumping station where it will be pumped into a holding tank. In the holding tank, you will have aeration, so there will be what are called spargers at the bottom that will inject air, not directly oxygen, but just air. Air is much cheaper than oxygen through the holding tank, and then you settle out the solids. Then this will run through a sand filter in order to get rid of particulates, and then an activated charcoal filter to get rid of odor and any color. And then it's chlorinated, and then it's treated, and then that will directly go to the consumer. Okay. In terms of sewage treatment, that's a little bit different. So you start out with raw sewage, and this can include different types of waste, and you decant off the liquid residue, the liquid is going to be recycled, and then the solids down at the bottom then are collected for landfilling and disposal. Okay, and then the liquid, which is pretty raw at this particular point, goes through a sludge digester. Okay, and the water is then decanted off, and then the sludge itself is mixed and aerated, and then it is either disposed or reclaimed in what's called an anaerobic digester, where the solid content becomes even less. Okay, and once that solid content becomes even less, then the solids are decanted off or disposed and landfilled. Okay, and then the water here is taken over and it's filtered, it's chlorinated, and then that's released. It's not released directly into the water supply. Sewage, sludge, and wastewater that comes in is not appropriate to go to the water supply, but it may be released into a reservoir or a lake or a river. Okay, but it has to be treated first. So you have primary, secondary, and then tertiary water treatment. Okay. You also want to monitor water to make sure that you don't have different diseases in there, waterborne diseases. This would include Giardia and Cryptosporidium. Some of the bacteria would be Campylobacter, Salmonella, Shigella, Vibrio, and Mycobacterium. All can be transported in water. And then also hepatitis A and Norwalk virus can be transmitted through water, and this type of virus can be chronic and can cause many, many different health conditions. Okay, and then you also do testing based on fecal levels. Okay, so where you find contaminants, and then you actually want to see the number or count of fecal bacteria that is in a particular aquifer. And then the state of California sets standards for these aquifers to let you know, is this water safe for drinking? Is this water safe for recreation and swimming? Or should the water be avoided at all costs? We've run these different tests at Simpson University. We actually determined that the ponds were safe for swimming, although I still wouldn't recommend it. We looked at fecal levels and we found that they were in the acceptable range for recreational water, but definitely not drinking water. Okay, and then you want to look at water quality, and you do this by measuring the amount of dissolved oxygen in the water and the amount of dissolved oxygen that it takes to degrade any contaminants in the water. Okay. And then fecal coliforms, yes, I'm sorry to say that there are fecal coliforms in drinking water. The municipal drinking water standard is about 4 colonies per 100 milliliter of drinking water. So that would be 4 bacteria in 100 milliliters, which is about, a typical drink is about half a liter, maybe a little less. So that would be around 20 colonies that you would consume from municipal drinking water. Okay, this is a coliform test. The coliforms come up on this particular test plate after the water is filtered, and each one of these blue colonies represents a single coliform. Okay, in food, we have both beneficial and detrimental microorganisms in food. Fermented foods can improve the flavor or the texture. So things like sauerkraut or yogurt or cheese have active cultures in them, and then that can create a different flavor or texture for your food. And then there are some microbes, especially yeast extract, that serves as food directly. You may have heard of Vegemite. You may have even tasted Vegemite. I think it's just awful, but it is a food substance that is basically the extract of yeast. Okay, it has a really, really salty, almost petroleum-y taste to it, but it is a yeast extract. It is microbes directly. Okay, detrimental. This would include organisms that cause food poisoning or foodborne illness, and this would be microbes that also spoil food. That they may be safe to consume, but they're going to be undesirable. They're going to give it bitter taste. Cause the food to smell, or cause the food to change color, like mold in bread. Okay, and then there are some microbes that are neutral. They just will maintain small quantities of the microorganisms. This would be in different types of dairy foods, different types of breads, and also even meats. But they're neutral microorganisms, and usually they do not overgrow. As far as food fermentation, usually food is fermented with some type of starter culture like kombucha. And this just has the right mix of organisms for a given type of fermentation. Okay. An example of this would be leavening of bread with baker's yeast. Okay. And when you bake the bread, you actually kill the yeast. You're not consuming live yeast. But when the bread is being leavened and the dough is rising, then you're introducing carbon dioxide, which makes the bread nice and light and fluffy. Beer for ethanol formation has brewer's yeast in it that will convert the carbohydrates in the grain to ethanol. The barley also has to be malted, and that creates nutrients for the yeast. Okay, the malted barley is then ground into a mash and heated, and then the clear fluid then is boiled for 2.5 hours with hops to give aroma and flavor. Okay, fresh green beer is lagered. That's where it's cold aged, and then usually it's cold filtered after that, and then you get your final beer. In terms of wine, it's a similar fermentation process, but instead of using grain, you're using grapes primarily, although you can make wine from other types of fruit. And then liquors are basically first fermented and then distilled after fermentation to get a higher alcohol content. Okay. Vodka is distilled from potatoes. Rum comes from sugarcane, like rum from Jamaican rum. Rye comes from mash or corn mash. And brandy is actually a distilled wine. So you take wine and you distill it further to increase the alcohol content. Okay, so here is just a quick overview of how wine is made. So you harvest the grapes, you crush the grapes so you can get out the liquid. You may leave some of the skins in, which are rich in tannins, which will change the color of the wine. You get rid of the discards like the leaves and stems and things like that over here. You ferment the wine in the presence of yeast over a long period of time. And then you press the wine out, okay, getting rid of the yeast. You don't want the yeast in the wine. You allow that to age further. And then you finish it, and you might add a sulfiding ingredient. Sulfiding ingredients basically will prevent spoilage of the wine over a long period of time and interact with the ethanol directly to prevent spoilage. And then finally, you bottle the wine. Sulfiding agents aren't necessarily the best thing for you. Sulfiding agents for me give me a screaming headache, so I know if a wine has sulfites in it just almost immediately. Okay. In terms of milk and dairy products, microbes aren't really, for milk, they don't really enhance the taste of the milk. They can just cause spoilage. Okay. But milk is really the perfect media for microbial growth. It has carbohydrates in it, like lactose. It has protein in it, and it has a lot of fat in it. And so this enhances microbial growth. Now for cheese, you usually come up with a starter culture or rennin, which is a type of enzyme that you find in the stomach lining of cattle. Okay, and the rennin enzyme will promote curd formation. And it's very, very rapid. You want your curd formation to be rapid because if you let it go on too long, then the culture starts to take over. Then you separate out the curds from whey. And the whey is just the protein, kind of liquidy protein that's left behind. And then you allow the cheese to ripen. Okay, and when you allow the cheese to ripen, depending on how it ripens, then it will have different qualities. If you let it ripen longer, then the cheese is going to get sharper and have more of a more of a distinct taste. If you let it age for a short amount of time, it's going to be more mellow. Some cheeses require additional microbes that will be introduced during aging, like Roquefort cheese or blue cheese has a mold in it, and other yeasts and micrococci and molds are used for aging cheese. Okay, here is a cheddaring table where they're actually taking the curds and dividing them from the whey. Okay, here's after the curds have been packed, and they'll be packed into containers for aging after a while. And then you get a cheese wheel. Okay, so they're packed in these containers and then they're aged, usually in a refrigerated environment for a period of days or weeks. Okay, I did go to the Tillamook cheese factory a long time ago, and I saw individuals working with the curds and on the cheddaring table, packing the curds together. And one of them wiped their nose, and then after they did that, they stuck their hand directly into the cheddaring table. So that's where they get the yellow color. No, not really. Sorry. Okay, I just had to add that. Just make sure you're awake. Okay, some microorganisms serve as food, like Vegemite is just a yeast extract. Okay, an animal food feed called protein comes from Methylophilus methylotrophus. Mycoprotein from Fusarium, and spirulina. When you see spirulina in health food stores, that's directly you're consuming microorganisms. And here's Vegemite. You can buy Vegemite sometimes at different food stores in the United States, but mostly it's associated with Australia. You can take it and mix it with cheese for a cheesy bite. Okay. And then we also have to think about microbes in terms of what type of foodborne diseases they may cause. Okay. And so food poisoning is pretty common. Several million people suffer from food poisoning or food infection every year. You have to be very careful with it. You don't want to just run its course. You may need to get it diagnosed. It could be something that's bacterial, like salmonella, a pathogenic strain of E. coli or Vibrio, Listeria, Campylobacter, or it could be something that's viral, like hepatitis A or Norwalk virus. Okay, there's a lot more outbreaks. Even though the population hasn't doubled, outbreaks have doubled over the past 20 years. And that's probably because of the advent of more fresh processed foods. Things like bag salads are notoriously bad for food poisoning because you have individuals that are working in each link of the chain and they may have poor hand washing practices or they may have touched the doorknob or something like that and not washed their hands. And so that's when you get contamination of these foods. Since they're not cooked, the bacteria are not sterilized and then they're passed along. So things that are packaged that are fresh are notorious for food infection, and so you have to watch those very carefully. Okay, so processed foods, raw vegetables, fruits, and meats, things that are not cooked, can really be a problem. And there is what's called the Hazard Analysis and Critical Control Point Program that helps isolate where did the food infection come from so the food can be appropriately recalled. Okay, and this just shows different over the past years, past 20 years, how different infections have increased or decreased. I don't really like that slide. Okay, it's a little confusing. So you want to prevent food spoilage and obviously food poisoning. You don't want to get people sick, and you also don't want your food to spoil. So first of all, you can just prevent incorporation of microbes in the food by having a clean environment where you're cooking. You also want to prevent the survival or multiplication of microbes in food. So you can use temperature, okay, heat sterilization or pasteurization. Pasteurization does not kill all the microbes, but it kills the majority of the storage microbes. You can also monitor cooking temperature, or you can refrigerate or freeze food so they will last longer. Okay, so we start out with our beef here. Individual that is butchering the beef has good hand washing practices. So you can use heat, like canning of food or pasteurization or cooking of food. You can irradiate food with ultraviolet radiation. You can filter food with a submicron filter to get rid of bacteria. And then you can maintain temperature. You can cool it or freeze it, or you can heat it up. And then you can add different preservatives like salt. Sugar can be a preservative in very high quantities. Nitrates, although nitrates can cause cancer, so you have to be very careful with that. Okay, and you have to avoid the hot zone in terms of where bacteria multiply. If you're looking at physiological bacteria, most are going to multiply very rapidly between 98 and 100 degrees Fahrenheit. Bacteria are going to multiply between, probably between 80 degrees and 104 degrees Fahrenheit. And then bacteria are going to multiply at a reduced rate from about 50 degrees Fahrenheit all the way up to 145 degrees Fahrenheit. Okay, so you want to avoid body temperature. And so bacteria growth is inhibited by refrigerating and freezing, or you can destroy bacteria by getting close to the boiling point. Okay, UV is used for surface sterilization. Say, if you're working with a surface where you're preparing food, then you might treat it with UV light for several minutes before you start to prepare the food. Food is gamma irradiated to get rid of things like bacteria and worms, and it's very prevalent. It's also an area of controversy because of irradiation changing the food. You can add salt or other types of preservatives, organic acids, nitrates, nitrites, and sulfites. Very high sugar like syrup. That's something that bacteria are going to avoid. You can dry it, or you can actually use bacteriophages that will fight off, these are viruses that will fight off the bacteria. Other microbes are used for good production, okay? Things like organic compounds, succinic acid, lactic acid, acetic acid. This could also include antibiotics, hormones, different vitamins, acids that I mentioned before, solvents and enzymes. Okay. And when we look at microbial growth in industrial settings, we can look at both primary metabolites. These are things that are essential to microbial function, as well as secondary metabolites, that these are not critical to microbes' function, but they may serve a purpose in society. Okay. And so usually, if you look at the lag phase of any microbe, then there's not a whole lot of growth going on. It's just processing the substrate, assimilating enzymes to break down the substrate. Then it goes into the exponential phase, and this is where you get your primary metabolism. This is where the microbes are producing things that are essential for it. And then in the stationary phase, then you get more secondary metabolites. These are more byproducts that aren't necessary for growth, but they may be breakdown products of the cell. Okay, so we can look at that and we can start to use industrial tricks to get secondary metabolism. So say if we want to make penicillin, then we may add lactose, which is going to cause slower growth, but more penicillin formation when we're cultivating the mold penicillium. And then we may select microbial strains that have been mutated so they don't have any product feedback inhibition. Some products can inhibit the growth of organisms, but you can reduce that by mutating the organisms selectively, so you can increase the type of product that you're trying to produce. You can also use genetic engineering and biotransformations. And so tetracycline, you don't get from a single organism, you get from multiple organisms, and it takes 72 different chemical steps in order to make it. Okay. Here's just a bioreactor. You can see that they're adding nutrients in. There's a cooling jacket because microbial reactions usually generate heat. And you can sample the product, and then down at the bottom there's an air sparger, and you'll just let this reactor run through its lag, exponential, and stationary phases, and then you'll harvest the product at the bottom. You can also have more of a complex type of fermentation where you recover microbes. You may recycle microbes as an inoculum. Then you recover the product below, purify the product, and package the product. You can actually collect and recycle the medium as well. So this is more advanced. This just shows how you can use a fermentation, recycle the organisms, recycle the medium, and then come up with a product at the end of the line. Other types of products, Bt crystals, Bacillus thuringiensis, is a natural insecticide. It's toxic to insects that attack potatoes, corn, rice, and wheat. Okay, and then other products include enzymes that could be used for manufacture of chemicals, for agriculture, for food processing. There are enzymes that are added in order to increase the nutrition of food or increase the taste of food, make it tastier. There are enzymes for the textile industry, and also for the cleaning industry. There's a lot of enzymes that go in detergents, pulp and paper formation, and then dry cleaning. Okay, and then finally, a lot of ACEs, proteases, amylases, lipases, oxidases, and cellulases are all used in processing of different products. And then amino acids that are used for increasing nutrition, organic acids, solvents like alcohol, and even acetate that can be used as industrial solvents, and then also natural flavor compounds. All can get from microbiology. Okay, and that ends this particular session. So we'll go ahead and process this and get this up. And then on Thursday, I will have a mini lecture. I haven't decided what I'm going to talk about yet on Thursday. I might talk a little bit about the coronavirus, but then we will have a review, an hour-long review for test 3.